Welcome all to the presentation on the topic whole organism vaccines. So first we can see what is whole organism vaccines. Many of common vaccines currently in use of consisting of inactivated and live but attenuated bacteria and viral parasite, which mean whole organism vaccines, which means an organism that completely used as a vaccine against a particular disease, that is whole organism vaccines. These organisms may be bacteria, virus, or other parasites also. So first we can see the attenuated viral and bacterial vaccines. So there is an example which is a bacterial vaccine is Bacillus calmette-guerin (BCG). It is a bacterial vaccine that is obtained from the bacteria Mycobacterium bovis. A medium containing increasing concentration of bile. After 13 years, this strain had adapted to grow with increased bile and become the sufficiently attenuated and stay suitable as vaccine for tuberculosis. So, this is a method for preparation of the BCG. First, this bacterium that is culturing in the medium of high concentration of bile, and this vaccine is suitable for the disease tuberculosis. Next, we can see a viral vaccine, that is Sabine polio vaccine, which is obtained from the virus poliovirus. Virus attenuated by growth in the monkey kidney epithelial cells. 3 strains of poliovirus administered orally to the children on a sugar cube or a sugar liquid. So, this vaccine is this vaccine that is cultured in the monkey kidney epithelial cells. There is 3 strains of this polio vaccines which is administered to children orally. So we can see detailedly. Virus colonies in the intestine and induce protective immunity. This is the process how the vaccines that making immunity. So first, virus colonies in the intestine and induce the protective immunity. It induces production of secretory IgA, defense against naturally acquired poliovirus, also induce IgM and IgG. IgM and IgG as immunoglobulins. And it requires booster because of each strains interfere with each other's replication in the intestine. So this vaccination need 3 strains. 3 booster, which means 3 strains. These 3 strains completely destroy the disease causing virus. So we can see the 3 strains, about the 3 strains. Okay. In the first immunization, which means first strain, one strain will predominate in its growth, inducing immunity to that strain. In the second immunization, immunity generated by the previous will limit the growth of previously predominant strain in vaccines, enabling one of two strains predominate and induce immunity. In the third immunization, immunity that provide to the all 3 strains. This is the 3 booster of polio vaccination. Actually, all attenuated vaccines are of single booster, but in the case of polio vaccines, it is 3 strains. So completely, this 3 strains, after these 3 strains only the third immunization and the complete immunization complete. Next is rubella virus. Strain of rubella grown in the duck embryo cells later in the human cell lines. So another examples. Next we can see another type of vaccines. So first we see the attenuated bacterial and viral vaccines. Next we can see the parasitic vaccines. So parasitic vaccines. Parasitic diseases be a major obstacle to the farming, health and reducing the animal productivity. So, vaccination is considered one of the best alternative for the control of the parasites in future. In order to develop a vaccine, need to identify the target antigens involving the salivary gland antigens, secretory and excretory antigens of helminths, etc. are prepared from it. Vaccination developed for three major parasitic diseases, that is malaria, leishmaniasis and schistosomiasis. The difficulty in developing vaccines against parasitic diseases identify an appropriate antigens because of the lack of complete understanding of the type of immune response required for the protection. There is some examples, okay, of parasitic vaccines. First one is Theilaria annulata. It is a parasite, and from this parasite attenuated cell line vaccines can be prepared. And another one is malaria. This vaccines host is human, and this vaccines against it is mosquitosis. So next is combination vaccines. Combination vaccines are mixture of individual vaccines before administration. Some vaccines for an individual disease are multivalent in that particular pathogen has multiple types. It is referred as the multi-type combinations, which are same design and mixed together at a time of manufacturing into a combination vaccine. So, combination vaccines are of two types. First one is multi-type combinations, which means the individual disease are multivalent in nature. So other combinations are mixture of vaccines already licensed for different diseases such referred to be multi-target combinates. The development of combination vaccines begins with the combination of individual diphtheria, tetanus, pertussis vaccine into a single product. So we can also see another examples of combination vaccines, that is MMR plus varicella. It is against the chickenpox. Prevent 4 disease, measles, mumps, rubella and varicella. Next one is hexavalent vaccines. It consists of DTaP, Hep B, IPV, H1B. To protect kids from the diphtheria, tetanus, pertussis, hepatitis B, hemophilus influenzae and polio. So, this is all about the whole organism vaccines. First, we see the viral bacterial vaccines. Next, the parasitic vaccines for the parasites. And after all, the combination vaccines, it is consisting of more organisms in a single use more organisms for a single vaccines, or it defends against more than one diseases. So that's about all about the whole organism vaccines. Thank you.